again for the camera. What's your name? Morris?
My name is Morris Chuckman. Graduate student at the University of Illinois at Chicago in bioinformatics. That's a bioengineering, kind of a class, category of bioengineering.
How'd you get interested in this?
I don't remember. Let's see. Well, I was doing my undergraduate work in computer science, and I just kind of got I don't know. I just had some friends that were into organic and biochemistry, and they went off and worked at pharmaceutical companies for a while, and told me how this wonderful field is kind of emerging. And
well,
you know, I think so. I've seen some pretty good things come out of this field. I don't know, lots of nice little cancer therapies and things like that. Still growing,
still things to do.
Oh yeah, there's lots of we're just kind of, we have so much genetic data that we're kind of
repelled by the water, so that they will
be pushed into and packed together into the center of the molecule, giving it its final shape. And so all of these forces have to pretty much be engineered, so to speak, just right by nature and the evolutionary processes that pick which amino acids go where by field that's setting, kind of centered around organizing all this information. Because I mean, it kind of looked like we were using some sort of a microscope or something, but this information is actually kind of one of our. It's kind of it's all computation. It's all kind of like computer graphics and simulation. There wasn't any actual experimentation involved. Yes, this was all computer generated. I mean, these. For example, this protein here was never the procedure that's usually implemented. That's usually gone through to get the atomic coordinates of a protein, of a molecule this big, is usually very, very expensive, and the world can only probably do about 7 of them on a good day. You have to take the protein, you have to cool it down. You have to, well, first you have to purify it from the DNA templates. But there's other proteins that actually go around. They are genetically engineered in this way to bond to DNA and actually make more protein off of that DNA template. The sequence of the DNA is translated into the sequence of amino acids that coils up in this particular shape. Well, not this particular shape, whatever particular shape is that is useful for whichever piece of DNA is being translated, I guess. And about 90% of the DNA in an organism is never translated anyway. It's genes are dead. Lots of computation is involved in reassembling that pattern back into the three-dimensional coordinates of all the atoms in the protein. So these things are very expensive, and the size of the amount of lasers and liquid nitrogen and stuff like that is, you know, pretty much need a national lab to do this. Zoomed in on it, there'll be like a little V where the kind of the 2 points of the V are hydrogens and the nexus is the oxygen. But I mean, you can really see, like when it's spinning around like this, you can see it kind of as it would exist in nature, with kind of a little micro droplet of water around it, and So in the very center of this would be all of the amino acids that are that are repelled by water, and these outer ones are the ones that are easily dissolved by water. And I actually clicked on some of these amino acids and their names have popped up. They're spinning around with them.
But the names are spinning around?
Well, that basically is like if you take all of the little X and Y shaped things in the chromosomes and stretch them all out, and we have all of that genetic data. But again, we don't. This P73 occupies probably like a very small portion of all that data, and we can't. We haven't actually gotten around to actually crystallize. Which stands for monosodium glutamate. It's in position 248. OE1. I'm not sure what that means. But yeah, you kind of get a view of this, of the three-dimensional structure of the molecule and its native water encapsulation. Maybe let's see here. We'll do let's see, index charge. Color these guys based on their charge, maybe. They're all white.
What charge is?
It's the positive or negative.
Yeah, I know, but
oh, okay. Yeah, that's not going to work, I guess.
it's interesting shape.
Well, that's just all of the water molecules. I've just kind of visualized them in their space filling.
Oh.
Models. And, okay, you can stop. Stop spinning. Okay, let's do something a little bit less. There, we're kind of using our computer know-how to various tricks to simulate how this how it folds up into its three-dimensional shape by basically seeing that its sequence is similar to these other things, and those things have this shape. So it's just very exciting. There's lots of guesswork, but there's lots of potential rewards for guessing right.
Sounds like fun.
Cool.
Great.
Yes.
Okay, thank you.
You're welcome. Yeah, I guess you can't really see any kind of a rendering of this molecule through the water. That's why I took the water out in the other one.
Yeah, you can see sort of how these are like, they look like they're chained together.
Yeah.
Kind of interesting.
Yeah.
I've got it so magnified.
Oh, yeah?
Yeah.
Well, the individual water molecules are loose. They're just kind of rolling around one another, kind of attracted by their own